Hello and welcome to Cosmo Intel Research. This study has been done to research the effect of T consciousness fields on skin allograft survival in rats. This is Farnos Kodododi presenting this study. For all the resources and references for this research, please refer to cosmointel.com. Skin grafting is widely used in repair of skin surgeries. Since a critical issue related to an allograft is its rejection, it is necessary to find a method that can prevent allograft rejection and increase allograft survival without side effects. T consciousness is the fundamental element of the universe. There are various T consciousness fields, or TCFs, with non-physical entities that can be examined through laboratory experiments. The effects of TCFs are initiated through the connection to the Cosmic Consciousness Network CCN, which is established via the Faraday-Monger's mind, and with applying them, specific information is transmitted. This entire experiment was carried out as a double-blind method where lab technicians were completely unaware of TCF's theory, and the Faraday-Monger at the Cosmo Intel Research Center, who established the connection, was unaware of the details of the study. 20 rats were randomly divided into two groups, the control and the group receiving tacrolimus. Then the group receiving tacrolimus after surgery was divided into two groups. One group was affected by TCFs A and B, and another group was not affected by it. After 2 weeks, histologic and macroscopic observation were performed on all groups. After grouping, the laboratory animals were anesthetized, and after preparation for allografting surgery, a full-thickness dorsal skin, 2 cm diameter circle shape, was harvested from the donor rat, and then the skin allograft was transplanted on the back of the recipient. After 2 weeks of storage, tissue samples were taken. The results showed the transplant in TCFs-related rats was successful and the new epidermis was formed, and sebaceous glands and high number of capillaries could be detected in the dermis layer. The macroscopic evaluation of allografted rats affected by TCFs displayed that these rats showed better adhesion of the graft and hair formation on transplant skin around them, as you see in the figure. Here, A2O panel show the 2-week trend after surgery in the non-treatment group, where the graft area was not fully attached to the skin of the mouse. The skin graft was being rejected and no hair had grown on the grafted area. Histological features of grafted areas in the TCFs treatments group A shows the collagen fibers were thick, more arranged and denser. Granulation tissues indicated by a high number of capillaries. Mild fibroplasia or mild infiltration of fibroblasts and inflammatory cells shown here. In group B, the collagen fibers were thick, more arranged and denser. The inflammatory cells and reepithelialization and sebaceous glands were observed. Histological features of grafted areas in the non-treatment group A, the collagen fibers were thin and weakly arranged granulation tissue indicated by high numbers of capillaries, fibroplasia, or severe infiltration of fibroblasts and inflammatory cells. Also in B, the partial reepithelialization was observed in this group. Histological feature of the control group here shows the scab is formed on the wound that is healing, and below the scab is the formation of a reepidermis that is thickening, and below that is the granulation tissue that is being completed. Our results showed that TCFs had stimulatory effects on better tissue adhesion located at the skin graft site, which led to better skin graft performance. This observation suggests that TCFs can be considered a promising option to promote the viability and integration of skin allografts. Although we cannot measure TCFs quantitatively, it is possible to investigate their effects indirectly through various experiments. Further research is needed to clarify the alleviative effects of TCFs on graft survival.